Alright, so here we are, video 1 of our coronavirus closure. We're beginning chapters 11 and 14 over genetics and human heredity. The way I've done these notes is I have uploaded to the team's assignment your notes, and you can print your notes out, you can handwrite these. I realize now I just put up the wrong notes here, so give me a second here and get the right ones up. Alright, so we have the right notes up here now. So you can print these off yourself, and what I would probably do is I would watch the video, I would let each slide end, and when you get to the end of a slide, I'd write down all the information after I pause the video. That's the nice thing about the video part, you can pause it so that you can listen to what I'm saying and watch what I'm doing and not worry about writing, and then pause it and then go in and write on all the information as it's up on the screen there. It's a good way to do that. It might be superior than actually live. So our attention today is we're going to be going through parts A and B. So those are the parts we're there for. You're also more than welcome to write these notes on the digital version. And to remind you how to do that, you got to open them up into Desktop Word. I have Desktop Word up here. You click on the Draw menu. You guys have touchscreen devices, so you can touch that with your finger. You can click on the pen. You can change the pen type, and then you can physically write in different stuff. And I messed up there. So if you do write, there we go. And so you can draw stuff in here and you can write like that or so. So that's up to you, whatever way you learn best. I would definitely somehow record these notes though. I think that's important part of your learning. Okay. What are you going to be able to do at the end of this? Well, we're going to be talking about human heredity, or heredity in general, and then human heredity. So we're going to be applying rules of dominant recessive traits, which you've had before back in middle school. You're going to be able to interpret a karyotype, which is our first mission here today. You're going to perform 1- and 2-trait Punnett square genetic crosses, which you've done what's called monohybrid crosses with 1 trait, and we're going to be doing something with 2 traits, which is called dihybrid crosses. Di meaning 2, so we got 2 different traits we're looking at. The 2 traits will be new to you. We'll be using Punnett squares to interpret codominant and incomplete dominant traits. And at the end, we'll be using pedigree charts to interpret human inheritance patterns. And finally, explaining a process called meiosis, which is similar to mitosis, that creates 2 gamete cells. So, the chromosome. You've seen these before. We brought these up. And the way the chromosome works, you can see these little X structures. So this whole thing here is the chromosome. And I've got an electron micrograph picture here, and I've got over here on the right just a simplified drawing. The DNA is wound up tightly, and these little proteins, they're wrapped around. You can see all these little green balls throughout here. These are called histones. Now the histones are what we had to break down when we had to extract our DNA from our mouth cells using the Gatorade, if you got a chance to do that. So the chromosome is just a thread-like area where DNA is wound up tightly in the nucleus of the cell. But here's where your genetic information is coming. So these little thread-like strands are the DNA. All right. So a little bit more into the chromosomes. Humans have 23 pairs of chromosomes. So 23 times 2 is a total of 46 different chromosomes in humans. Now 46 is not a lot. There are actually plants that have thousands of chromosomes. So there's no relationship between chromosome number and like how advanced an organism is, so to speak. So you don't have to have very many chromosomes. There's some worms that I know have 4 chromosomes or so that are used in research because they're easy to study. So the picture over here represents 2 chromosomes, and we got the whole chromosome over here, and you could call each side a single chromosome, but more specifically they're known as chromatids. So each side is a chromatid, and when they're joined together at the center, which is called the centromere, they are a single chromosome. You'll notice there's a specific banding pattern on there. You got black, white, black, white, black, white, and they're different sizes. And what that there is to represent is to illustrate to you that different regions of the chromosomes have specific different traits on them. These are called genes. Now, these genes are a lot larger on the picture than they are probably in real life. So each chromosome could have maybe thousands of genes on it, but on this one I'm only representing like what, 15? 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13, 14 of them. So just a simplified picture there. Now, these genes are equivalent. So this big black gene down here codes for the same thing. The little white gene codes for the same thing, etc. So what we do with chromosomes is one of the things we can do is we could look at genetic diseases using what's called a karyotype. And a karyotype is an orderly arrangement of chromosomes in an organism, and we're going to be specifically focused on humans instead of a lot of other things. And you can see here the chromosomes are arranged in certain ways. You notice that the biggest ones are over here at the top, so it always goes largest to smallest, and you physically measure them on a microscope, and you measure how long the chromosomes are. The first 22 of these chromosomes are called autosomes. That's where the vast majority of your genetic information comes from. The last pair may or may not match are called sex chromosomes. And these sex chromosomes are what determine the difference between the sexes of at least mammals. Other organisms have different ways to determine sex, but at least in mammals, it goes on an XY chromosome pattern. Now, if you have two X chromosomes, so here's the X chromosome right here in pink, you're a female. So all of you females and all of your cells, you have two X chromosomes. And if you're male, you have an X and a Y. Now, the X is usually larger than the Y chromosome. As a matter of fact, it's not usually. I think it's all the time, at least in humans. I don't know about other organisms. But the X is larger than the Y chromosomes. And there's some genetic information on here that codes for different things besides just sex. But a lot of your sexual traits are coming from these particular two chromosomes. All right, so the nice thing about karyotypes that you can do with them is that once you lay out the chromosomes, there's actual diseases that can be associated with different types of mutations in the chromosome arrangement. And so karyotypes are used to detect some genetic diseases. The picture on the right here is an example of a karyotype with 3 different genetic problems. I won't call them diseases because they may not lead to actually any disease. So I want you to pause the video and take a look on the karyotype here, and I want you to see if you can pinpoint the 3 mistakes, and then after you pause the video, we'll start back up and talk about it. So I'll give you a little bit here of time to look at it. So I hope you had time to pause and take a look at the karyotype mistakes on here. So the first mistake, I hope you found number 2 right here. If you take a look at it, there's a missing portion of the chromosome. This is called a partial deletion. And the partial deletion probably doesn't lead to any major problems because you have 2 copies of your DNA. So the other chromosome has the other copy of your particular DNA trait. So those genes aren't missing. You just got 1 copy, and all you need is really 1. If you found the second one, this is going to be occurring on number 16. Right here, we're missing an entire chromosome. And again, because you have a second copy, a backup copy, this usually doesn't lead to any usual major problems. It might a little bit, but nothing major that I know of. And the last mistake that's on here, the last problem, is a chromosome number 21 right here by my mouse. And if you take a look at that, there's 3 copies of that. This is called a trisomy. A trisomy is a 3rd copy of a gene. And I put a little skull and crossbones on here because usually a trisomy is lethal. Like if you get trisomy 1, 3 copies of chromosome 1, or trisomy 4, or trisomy 7, those can be lethal. Lethal to the point that when the sperm and egg cell meet and begin to divide and multiply their cells, that's inviable. When egg and sperm meet, it's called a zygote, and that zygote is not going to rise to a viable fetus and is removed and gotten rid of, and sometimes people don't even know they were ever pregnant. So, usually very dangerous. Trisomy 21, though, is not lethal. This leads to a condition that's well known called Down syndrome. Many of you have heard of that before. All right. I think this ends the first video. So if you go on Teams, I got a set of questions for you to answer, and you can complete those about today's lessons. And then tomorrow, you're going to be doing an online karyotyping lab that I found. All right. Thank you very much.